have a stylus to write on the screen, so we're gonna do it the old-fashioned way. And, but I'll still take pictures of these pictures that I draw, and I'll post it as a PDF. Before we begin, like 1 minute, just the syllabus. I know you guys have had it. I just want to just talk about just a couple of things, how you get in touch with us. I mean, I have my appointment there. I am really, really accessible. All you got to do is say, Dr. R, can we meet on this day, this time? If I can do it, we will. If we need to figure something else out, we absolutely will. I respond to emails a lot of times in minutes. I always have my phone with me, no matter where I am. I get my emails on my phone. If I don't respond to you within 24 hours, I missed it somehow. And so email me back, all right? So, and don't be afraid to. So just Dr. R, you might have missed my email, and so this is my question. Todd, I'm fairly sure that he's fairly accessible as well. When it comes to the materials, you can get that all free online. Exams, there's gonna be 4 of them. It's, again, as it says, mostly multiple choice, some short answer. When it says essay, we're not talking essays this long, we're talking essays this long, all right? We don't want to read essays this long anyway. And what you're gonna get in med school, which I think everybody in here wants to do at some point, it's all multiple choice in med school. So we'll try to gear this course as close to med school as possible, although in grad school, every once in a while, we do the short answer essay thing. When it comes to your grades, what you need for A's, B's, and C's and such, not that everybody wants a C, what you need for an A is at a minimum 394 points. There might be a slight tweak at the end of the semester. It'll just depend on how the entire class is done. And then as far as what your life is gonna look like for the next 8 weeks, well, that's what your life looks like for the next 8 weeks. R is for Tucci, M is for Natat, and we tell you exactly what's gonna happen. We might actually be able to get done with the endocrine system by Wednesday, although this is going to slow me down today. So I might have to peek in here a little bit on Friday, which is in the syllabus itself, but I remember last semester I got it done in 2 lectures. And so that's really it. Really, really straightforward. We'll try to make this as entertaining and educational as possible. When it comes to questions during the exam, or during the exams, if you do have questions, you can certainly ask us. But during lecture, just don't even have to raise your hand. Just if there's something I'm talking about, you want to have a question about it or add something to it, please feel free. I'm sure Dr. Mattel's the same way. Really laid back in here, I promise you. I know that I have a kind of that RBF, that resting bitch face, but I really am a laid-back guy. I promise you that I am a very, very approachable. As soon as you interact with me, you're going to find out soon that that's going to be the case. All right? Okay, let's get started. I think I probably had at least most of you in that neuro class, and so you know that I draw a ton. And so that's what's going to happen. That's why I needed this paper up here, because we're going to draw. And so the first drawing of the semester is going to be an overview of the endocrine system. And so I'll take my fancy pencils here, and we'll draw our first picture. So we're going to draw a gland, and I'll label it. I'm not used to switching back and forth. It's usually always on. If it's not on the screen, just say Dr. R. Switch, please, or whatever, we can't see it. All right. And I didn't even do my nails today. All right. Okay. Okay, so this is going to be a gland. And this gland is going to produce some hormone. And we'll just label it gland. Now, it doesn't necessarily have to be a gland with epithelial cells. There are structures that we're going to be discussing that's made of endocrine tissue, or neural tissue, but we'll get to that when we get to that. So that gland is going to produce a hormone, and we'll make the hormone a certain shape, and so that's going to be our hormone. And so I'll just label that hormone. And that hormone is going to be released by that gland, and that hormone is going to go to a cell, a target cell. And what that gland wants to do is tell that target cell what to do. So this is going to be a way that this gland, the epithelial cells of this, I bet just because it's doing this, huh? I don't know if there's going to be any way around that. I just hit auto. Yeah, that might have been the bad thing to do. Yep, that was a bad thing to do. I don't think that button did a damn thing. All right. Well, hopefully it behaves at least semi-okay. All right, so there's our gland and there's our hormone. Down here is going to be our target cell. Now, this target cell can be anywhere in the body. That gland can be anywhere in the body as well. And so we want to tell this target cell what to do, and we're just going to call it a target cell. And so how is the hormone going to get to the target cell? Well, it's going to get to the target cell because the hormone is going to be dumped into the blood. So let's draw the blood. And so this is going to represent the circulatory system, which you get lectured on with Dr. Natat later on in the semester. So there's blood vessel. So we're just going to simply release the hormone into the blood. Hormone is now in the blood, and then it's just going to simply circulate to our target cell. And that target cell is going to have a specific receptor on it. Now, this receptor could be in the membrane, this receptor can be in the cytoplasm, that receptor can be within the nucleus itself. I'll just put it on the membrane for this particular picture. So now our hormone is here, and then it's going to bind to that receptor. If it doesn't bind to the receptor, the hormone might as well not have even been there. We need that receptor to be there in order for that receptor to be then activated. So we activate the receptor, and then there's going to be some effect within the cell, and that effect is just going to be a cascade of reactions. It's a lot of biochemistry. We're not going to get into a lot of the biochemistry. We'll get into it a little bit. And then there's going to be some appropriate output or some appropriate effect. And so now we have this communication between, again, the cells of the gland and whatever this target cell is. We call this endocrine signaling. There are a number of criteria that hormones have to meet in order for them to be considered a hormone. One of them is to be dumped into the blood and go to some cell removed from wherever that hormone is being released. And so this is endocrine signaling. This is this chapter in a nutshell. And so what we're going to do from this point forward is going to detail about these kind of things. And what we're going to spend the most time on is the effect. That's what we're going to spend the most time on. We're going to talk about a hormone and what its function is. We're going to do a lot of pathophysiology as well. And that always helps us with physiology. And pathophys is, well, what medicine is in the end. That's why patients come and see you, because something has gone wrong. We'll certainly do what happens when things are good, but we'll certainly do a bunch of stuff when things have gone wrong. And so that right there is just simply all those words right there. So a very simple picture, just to kind of get our feet wet when it comes to the semester. Now, there's a bunch of different kinds of hormones. And these are the 3 different kinds. Now, what I want you to know is which of these is a protein hormone, which is an amine, and which is a steroid hormone. What I've done in this particular screen here, or this slide, these are all the steroid hormones that we're going to be talking about. Are those all the steroid hormones in the body? Absolutely not. But those are the ones that we're going to discuss. Every single one of them is right there. Every single one of the amine hormones that we're going to discuss is right there. Now, these are not all protein hormones. Why? Because the list was much longer. So if you know what the steroid hormones are, it's right here on the screen. If you know what the amine hormones are, it's right here on the screen. Every other hormone that we discussed is a protein hormone. So that's an easy way to remember them. Now, something else that's important to know is that the protein hormones and the amine hormones are water soluble, and what that means is that they can be carried freely in the blood. For the most part, there are some exceptions. And so the way I've drawn this picture is that hormone's not being carried by anything. So that's probably a protein hormone or an amine hormone going to its target cell. Now, if that was a steroid hormone, I would have had to have a carrier carrying that particular hormone. Why? Because steroid hormones, that's fat. They're not soluble in water. Half of your blood is water. And so they would need to be carried. And so that's, again, a semi-important point to that. Now, let's talk about the receptors now. We're not going to spend a ton of time on this. Again, it's a lot of biochemistry. Obviously, the receptors on the target cells, and that's what receptor I'm talking about these receptors right here. Well, let me actually label it. I didn't even label it, did I? So that's the receptor. And that's the specific receptor to the hormone. Every hormone has a specific receptor. Now, when it comes to the receptor, that's how we're going to get the signaling done within the cell. That's how the cascade of events is going to occur. You need to activate the receptor. If the receptor is not activated, you're not going to have any effect whatsoever in the cell itself. What else do we know, or have to know? There's specificity. Every hormone has its specific receptor. So an insulin receptor is not going to bind, let's say, a thyroid hormone. Just not the right structure. And so there must be specificity. And sometimes the specificity is screwed up, and we'll talk about that in just a little bit. And then these are two terms I'm sure that you're familiar with, agonists and antagonists, and we'll have examples of these. Here's another thing as well, is that I'm talking about these kinds of things right now in this particular chapter, which I will address from time to time. But Dr. Matat will talk about blockers and agonists, and I'll do it in other chapters as well. And again, I'm pretty sure that you guys are familiar with the concept behind an agonist and an antagonist. We'll find it, and specifically with receptors itself, an agonist is going to be some, and when we talk about these, by the way, it'll be some drug, some medication that you guys will someday prescribe the patient because you are treating some certain condition. And there are certain conditions where you need an agonist of a certain thing, or an antagonist of a certain thing. And we'll give you examples, not just in this particular chapter, but throughout the entire semester when it comes to that kind of thing. So a little bit of pharmacology, not much. It's mostly a phys course, but we'll throw in pathophys, we'll throw in pharmacology, a little bit of biochemistry as well. Again, to prepare you as well as we can for med school. So let's talk a little bit more about the receptor interactions. Here's another thing. I'm not sure what your background is, so I will always assume that if I talk about something in depth that you don't know what it is, if you do know the material, well, you're going to get it again. It's just kind of a reminder. And I'd rather err on that side to where I don't know if you know it or not. So, let's talk about interactions, once again, between the receptor and the hormone. It doesn't necessarily have to be a hormone, by the way. It's just I say hormone because this chapter is the endocrine system, but it can be a neurotransmitter, for example, that Dr. Matat might talk about, or that I might talk about later on in the semester. So, this is the way that it's going to work, and again, a very simple picture here. So we're just going to take a cell. This is going to be our target cell. And it's going to have the receptor, and I'm just going to put the receptor in the membrane. And so here's our receptor again. I'm going to not just put one receptor, but I'm going to put many receptors. So I don't know, I'll go with, just go 8. And you'll see why. Now when these hormones are binding to these receptors, the hormones that are circulating in the blood and making their way to the receptors in the target cell, not every single one of these receptors is going to get bound by a hormone. There's going to be a certain percentage of them. So 2, 4, 6, and there's 8. I'm just going to say that maybe 25% of these receptors are going to be bound by a hormone. And these are just normal conditions, by the way. So our hormone was green. So let's just say that these two receptors over here are going to get bound. And then there's going to be some effect within inside of the cell. So there's going to be a cascade, a normal cascade. And so that receptor is going to get activated and this receptor is going to get activated. And as a result of that, we're going to have a normal cascade within the cell. And when I say cascade, it's just some reactions that are going to occur. And I'll actually talk about them briefly in a little bit. Okay, second messenger cascades. And this is going to result in, once again, and I'm just going to say a normal effect. So you have a certain amount of these receptors that are going to be activated. Now, let's talk about when things go wrong, and when things go not wrong, but when things get better. You'll see what I mean by that. So we're going to talk about a mutation, we're going to talk about resistance, and we'll talk about sensitization. And so what we'll do here is just 3 columns, so a genetic mutation. And so a genetic mutation is simply, so those receptors right there had to be produced by the cell. And so there needed to be a recipe for those receptors. Now, recipe is a gene. A gene is a recipe for a protein. Those receptors are proteins. In order for those proteins to be present, any protein within the cell has to produce it. The gene has to have the right recipe. If it doesn't, we say that it's mutated. So if the recipe is bad, the receptor is going to be bad. And so now we're going to have a bad receptor. And I'll just put the word bad right there. Why is it bad? Because it was produced improperly because the recipe was bad. And what that's going to do is it's going to decrease the sensitivity of that cell to the hormone. The key is, once again, specificity. And so if the shape of that receptor is no good, because again, we have a genetic mutation, well, now the hormone's not going to bind to it properly, and now again, things are no longer going to be normal, and so now we're going to have a decrease in whatever effect that hormone is supposed to have. And we'll give examples of genetic mutations throughout the semester, and I think I'm going to give you one today as a matter of fact. So, genetic mutation, that's bad. Resistance. We'll talk about resistance in the course as well. I'll talk about it in the next lecture when we talk about insulin resistance, for example, a type 2 diabetic. And so over here, what's going to happen is in order for these receptors or cells to become resistant to a hormone, and that's what we're talking about, resistance to the hormone itself. So resistance to the hormone. The cell is now resistant to the hormone. Well, why would that happen? It's going to happen if the receptors are bombarded by the hormone constantly. And it doesn't necessarily, again, have to be a hormone, but it's the endocrine chapter, so we're just going to say it's a hormone. And so it's bombarded. The cell's bombarded, bombarded, bombarded. And so what can happen over time is that we get a decrease in sensitivity. Now, why are we going to have decrease in sensitivity? Well, there's a couple of reasons that can happen. What this can do over time is decrease the number of receptors. So decrease the number of receptors. If you have less receptors, well, obviously then you don't have the opportunity to have a normal cascade within the cell. So let's say instead of 8 that we have here, there's 4. And we still bind 25% of the receptors. Well, now we're down 50%. All right? So a decrease in the number of receptors can be something that occurs. You can also have a decrease in the shape, the structure of the receptors themselves. And so you have a decrease in, we'll just say we're going to change the structure of the receptor. Not because we genetically mutated them, but again, because of the conditions that we've had here. And so once again, both of those things are going to lead to a decrease in sensitivity and a decrease in the effect of the hormone itself. Now, something that can be done, and something that the body will try to do is increase that effect. Well, how can it do that? What the body will do is increase the amount of, in this particular case, hormone that's being released. So initially this can be overcome, this resistance that we have here. So initially, overcome with more hormone. But then over time, we're going to have a point of diminishing returns. So it's going to increase the resistance even more. Over time, it's going to make it worse. Oops. There we go. Running out of room here. So over time, it causes an increase in resistance. It makes it worse. But the body's going to give it a shot. All right? So that's resistance. So the first two, bad. Next one, good. And that's going to be sensitization. And so it's the complete opposite. With sensitization, and we'll give you examples of this as well, you actually get an increase in the number of receptors. And there are ways to do this. I'll give you an example in the next lecture. I don't think I give you one today. You change the structure of the receptor in a way that it's going to be, the affinity is going to be greater. Okay? So, again, where am I getting all this stuff? All this stuff is coming from this over here. So, here's our genetic. So that's what we just did right there. And now we're going to have what's called upregulation, sensitization, which is the complete opposite of downregulation. All right? And there's something else called the permissive effect, and I'll give an example of that as well. I know Dr. Mattal will probably give an example of that as well. So, increased sensitivity of the receptors, and what this is going to do is, well, now we have an increase in the effect. Oops, sorry. All right, so very, very straightforward stuff. And again, examples throughout the entire semester, not just this particular chapter itself. Now, when it comes to the receptors themselves, I already told you that they can either be in the membrane, or they can be within the cytoplasm, or they can be within the nucleus itself. It's going to depend on, obviously, the receptor. And when it comes to the plasma membrane receptors, which is what I have drawn so far, is that it's going to be protein and amine hormones. And when it comes to the intracellular receptors, binding steroid hormones and some amine hormones. So amines can do both. And so now we're going to do our cascade. And again, very, very simple biochemistry here. Very, very basic biochemistry. Again, I bet you most of you have had it, but I'm just going to remind you of it anyway. And so let's talk about these second messenger cascades. And these are going to be cascades where we bind a receptor in the membrane, and then stuff happens inside the cell that will eventually tell the cell what to do, what the effect is that we want. And so the first ones we're going to do are the G protein cascades. The G protein cascades are right here. And so let's just draw those very, very quickly. Don't need to spend too much time on this. So the G proteins. Now there's 3 G proteins that we're going to be discussing. There's more G proteins out there, but these are the ones that we're going to discuss. So, G proteins. So here's our target cell once again. And we're going to have our receptor here. I believe it's blue. And so there's our target cell. Oh, and by the way, I'm going to give you guys 2 breaks during this lecture at 45 minutes, and then another 45 minutes after that. So there's our target cell. Now, these G proteins are heterotrimers, and so they're made up of 3 different proteins in cells. And there are 3 subunits. There's a gamma, there's a beta, and there's an alpha. And so they're associated with the receptor itself. So that's the gamma subunit, then we have the beta subunit, and then we have the alpha subunit. It's the alpha subunit that's going to give the activity of that particular G protein. Now, there's 3 G proteins again that we're going to be talking about. So we have GS, we have GI, and we have GQ. And it's again the alpha subunit that imparts the activity of the G proteins themselves. And so S stands for stimulate. So that's going to be stimulatory. I is inhibitory. The Q is also stimulatory. And each of those is going to be affecting something else within the cell that's very, very specific. So what we'll do is, we'll do the GS first. And so let's talk about a receptor that is associated with the GS protein. It's going to affect an enzyme. So once again, we need the hormone to bind, by the way. That has to happen. That's how we're going to activate the receptor, which is then going to activate the G protein and the cascade that occurs as a result of that. So again, GS is first. And so here's our cell. There's our target cell. We need to bind, once again, the hormone. That's going to be number 1. And that's always going to be number 1. The first step has got to be, you have to bind the receptor. So number 1. Then what's going to happen is we're going to activate the G protein. And so we'll put over here, again, those subunits. So there's our gamma, beta, there's alpha, and I'm going to put an S right there. And so number 2, as a result of that, we activate the G protein. Now what? Well, there's going to be an enzyme. And this enzyme I'm going to put within the membrane over here. And that enzyme is going to be adenylate cyclase. And adenylate cyclase has a very specific job. But it's sitting there idle at this particular point in time. It needs to be activated. What's going to activate it is, once again, the G protein. Now, when we activate the G protein, what's going to happen is, literally, that subunit is going to move to the adenylate cyclase. And then what's going to happen is that adenylate cyclase is going to take ATP and convert it into cyclic AMP. So this is the cascade that I was talking about. So we're going to make more cyclic AMP. And that'll be step number 4. And then a bunch of other stuff happens within the cell itself to where eventually we get a certain effect that hormone wants that cell to do. So that's that. The GI is also associated with the adenylate cyclase, except the GI is going to inhibit it instead. And so we're going to have less cyclic AMP produced. And so we'll do that one really quick. So here's our cell. And we'll put our receptor there. I already know you know where this is going, but we're going to be thorough anyway. So the hormone binds, that's going to be step number 1. And then we're going to activate this particular G protein. Again, this is going to be a GI protein. So gamma, beta, there's our alpha, and we're going to put a little I right there. Number 2, we're going to activate that protein. But it's going to be an inhibitory protein. So even though we're activating it, doesn't mean that we're going to stimulate the cell. We're going to inhibit the cell because, again, it's associated with an inhibitory molecule. Again, we're going to put adenylate cyclase in the picture. This enzyme. By the way, it can be soluble as well, meaning it could be within the cytoplasm itself. I just chose to put it in the membrane. And so, number 3, same as before, the alpha I is now going to move. It's going to inhibit this. It's not going to stop the production, but it is going to decrease the production of cyclic AMP. So now we're going to have less cyclic AMP. That's still number 4. And then the GQ is going to be associated with a different enzyme. And this enzyme is phospholipase C, and so we'll do that one quickly as well. So what do you have to know from this? Just a simple cascade. S and I are going to affect adenylate cyclase. S is going to make more cyclic AMP or cause a greater production of cyclic AMP, and I is going to decrease that production of cyclic AMP. But now we have a different enzyme that's going to be affected by a GQ now. And this one's got a couple of extra steps. So we're going to put our receptor here. Again, that's always step number 1. The hormone is the first messenger. The cascade of events within the cell, like that cyclic AMP, is called the second messenger. So number 1, we're going to activate this particular receptor, which is associated with a GQ. So we're going to activate the G protein. And now we're going to put a different enzyme here. And this enzyme is phospholipase C. And so, 3, it's going to literally physically move, just that subunit. And now what's going to happen is that this particular enzyme is going to affect another enzyme called PIP2. So, number 4, I'll put a plus sign right there. So we have another enzyme, PIP2. And when PIP2 is activated, it's going to produce diacylglycerol and calcium. And so we're going to get an increase in that molecule, which is now the second messenger, and we're going to have more calcium. And then those things do other things within the cell. It's a black hole of information, as I like to call it. A bunch of biochemistry, but eventually that cell is going to do what it's supposed to do. And that's being dictated by that particular hormone. So again, very straightforward, second messenger cascades based on these G proteins. All right? So those three pictures that we just drew. Now in the notes I actually had IP3. IP3 is going to cause calcium to go up, so let me put IP3, I'll just be consistent with the notes. But IP3 will increase calcium within the cell. We'll just leave it at IP3 because I don't have calcium within the notes. All right? So that's those. Now we have other receptors that are not associated with G proteins, but associated with another type of enzyme. And so that's going to be guanylate cyclase. And so all we're going to have here is, what the hell, let's just be thorough. So we'll do the guanylate cyclase now. So this is not a G protein. This is going to be, again, so, cell. Receptor. Hormone, again, always first step. So there's our number 1. Now we're going to be associated with guanylate cyclase. And so the guanylate cyclase, I'm going to make this one soluble. So there's our guanylate cyclase. I'll actually go write the word guanylate cyclase right within it. And then what guanylate cyclase is going to do is it's going to take GMP, or E. What do I have in the notes? Okay. So then GTP. So number 2, we're going to activate that particular enzyme. Instead of ATP, it's GTP. And that's going to give us cyclic GMP, not cyclic AMP. We're going to have more of it. So that's going to be stimulatory when in its effect. So I put a plus sign here. Oh, I already put the plus sign there. And then again, other things will happen after the production of cGMP that causes the cell to do what it's supposed to do based on what the hormone wants it to do. And then we have other particular receptors that themselves are enzymes, like the insulin receptor, for example. When it binds insulin, it gets activated and causes some other stuff to happen within the cell. I'm not going to draw that one. All right. Now what? When it comes to, again, what we've just gone over, the receptors have been within the cytoplasm itself. What about those receptors that are in the, within the membrane? What about those receptors that are in the cytoplasm and the nucleus? Well, that's going to be our steroid hormones, and then again, our amines. Just there's a couple of examples with the amine hormones. And so we'll do that very, very quickly. And then we'll really get into our hormones, into the meat and potatoes of the chapter itself. Okay, we just needed to get this stuff kind of out of the way. And so I'll just do this in one picture. So again, we're going to have a steroid hormone, and this is mainly steroid hormone. And I'm just going to label that. That's a steroid hormone. Now again, there are exceptions to the rule, and we're going to be talking about those two exceptions when we actually get to the particular gland that produces the two amines, and they're going to be what are called T3 and T4. They're thyroid hormones. They too will bind receptors within the cell itself, but for the most part, it's going to be steroid hormones. So let's put our nucleus in the cell. We're going to put a sliver of DNA in here. And there are receptors that are sitting on the DNA molecule already, and so let's put one in there. So some receptors are already there. And what these receptors are going to do is activate or deactivate some particular gene. And so that particular steroid hormone, one example can be that it just makes its way all the way to the nucleus and binds to that right from the start. Or what it can do is make its way into the cell and bind to a receptor that's in the cytoplasm, and then that complex then makes its way into the nucleus and then binds to the DNA, activates or deactivates some particular gene. So that's one example, or another example, a second example. Or a third is, just makes its way into the cell, binds to some particular receptor within the cytoplasm itself, and that's where it stays, and then that's going to do something within the cell that the cell will then eventually do what the cell is supposed to do based on what the particular hormone wants it to do. So again, very, very straightforward. And then again, I'll just put it down here. T3 and T4, those are the thyroid hormones. We'll talk about it in a lot of detail in the next lecture. Also, bind to intracellular receptors. And that's going to be our exception, at least the one that we discuss. And so that's that. Again, just simple, straightforward stuff. Yes?
So steroid hormones don't always have to translocate to the nucleus?
Nope. Nope. All right. Now what? Now let's get into the endocrine system one. So, we're going to talk about a number of things here. First one's just going to be feedback mechanisms, and I know you guys know what negative and positive feedback are, but again, I'm going to visit them anyway. And so let's talk about these feedback mechanisms. Negative feedback is what we're going to talk about first, and that's what's going to be our focus in this particular chapter, is negative feedback. These hormones are telling these cells what to do, and in large part are telling the cells, the target cells, what to do to maintain homeostasis. And so this feedback mechanism, negative feedback, is going to be the most common, and so let's just draw negative feedback. So that right there, actually we'll draw an X and a Y axis. Oops, thank you. So we'll draw our X and Y axes. This is just going to be time. It can be time in seconds, minutes, days, it doesn't matter. And this is going to be the level of the hormone in the blood. So we're just going to concentrate on hormone right now. And whenever I talk about the level of the hormone, I'm talking about its level in the blood. And so that's going to be the normal level right there. But if you took blood work from a patient, and you took blood work at I don't know, 8 in the morning, did it again at 3 o'clock in the afternoon, did it again at, I don't know, 8 o'clock at night. It's not always gonna be exactly that value. It'll fluctuate a little bit. And that's what this represents. That represents the fluctuation around the normal level. There's always a normal range, and that's what that is. There's our normal range. So when you do blood work, when it comes to anything, level of sodium, pH, hormone levels, whatever, there's always gonna be a normal range. It's never gonna be, well, this is a normal value for this particular, whatever it is. There's always gonna be a normal range for it. And it's no different when it comes to a hormone. Now, when these hormones are within their normal range, they're maintaining homeostasis. Well, there's gotta be a way of keeping the hormones within a normal range. And that's what negative feedback is. And so I'll just make it a bit more dramatic. Let's say that the level starts to get a little bit high. We need to get the level back to where it is. Or, I'm sorry, back to within that normal range. That's negative feedback. Very simple concept. However, once we start to get into details of certain conditions and hormones, this really, really simple, basic negative feedback mechanism, sometimes it just kind of leaves us. And so you always want to remember the basics of things when you start to study the details of things. Because a lot of times when you get into the details of the material, you forget about the basic stuff, and it makes the details way harder to understand. If you understand the basics of things, the details tend to kind of fall into place much, much easier. That's also negative feedback right there. So negative feedback is just simply, if something gets too high, you bring it down. If something gets too low, you bring it up. That's all. And that's gonna help us maintain homeostasis. Why? If the hormones are maintained within a normal range, whatever they are regulating are gonna be normal as well. Now, it doesn't mean that hormone levels can't go up and down, above and below the normal range in order to maintain homeostasis within the body, because they're absolutely going to, and we'll discuss that. But then what must occur is that after those hormones have deviated from the normal range and fixed whatever they were supposed to fix, or maintained whatever they were supposed to maintain, then they should come right back down to within the normal range. If they don't, well, now that means is that we've lost negative feedback. And when you lose negative feedback, that's when bad things happen in the body, and that's when people come to see you to get fixed. And so that's that. Now, something else I want to point out that we're going to visit at a later time. So these hormones are regulated again in a negative feedback manner, and this is just what I just discussed right there. Now, these two lines right here, we'll talk about it later. Later meaning, I don't know, later on today. All right, not later on in the semester or later on my next lecture, but those two things we'll talk about today. Positive feedback. Positive feedback is a necessary phenomenon to facilitate what I like to call a special event. And so we'll just do positive feedback. So most hormones, all hormones I should say, behave in this particular manner. But there are a certain number of hormones that behave in a positive feedback manner under special circumstances, special events. And we'll see a couple of those examples in this chapter. Dr. Batat will talk about them too, with the menstrual cycle when he gets into the reproductive system. And so we'll once again do our X and Y axis. Again, this is time. Oops. And again, time, seconds, minutes, days, weeks, it doesn't matter. This is the level of the hormone. We have our normal range, so there's our normal level of the hormone, whatever the hormone is. And this is going to be our normal range here. Now, when it comes to a positive feedback mechanism, it's typically short-lived, meaning that initially, whatever hormone it is that we're talking about, we are within the normal range. But then, at this particular point in time, what's going to happen is that in order for this special event to occur, now what kind of special events am I talking about? Childbirth would be a special event. Lactation would be a special event, for example. So right there is the start of the birthing process. And that's the length of time of the birthing process right there. Or mom is breastfeeding the baby after the baby was born. Lactation. What needs to happen is there are certain hormones that need to go up, 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 well above the normal range for the duration of the special event, for the special event to occur. And so we're going to be up here. And that's completely fine. But then once the special event is over, which is right here at this point in time, well, then we're coming back down. Then negative feedback is going to take over. So this is positive feedback. What I mean by positive feedback is that the special event is feeding into the gland itself that's producing the hormone, and it's just making more hormone. And making more hormone and making more hormone. Today we'll go over a couple of examples of positive feedback. I've already mentioned them, birthing process and lactation, and the hormones involved with those two particular processes. And then Dr. Batati again will talk about a positive feedback mechanism in the menstrual cycle. But then once again, negative feedback takes over. The negative feedback has absolutely nothing to do with the special event. It's just, once the special event is over, negative feedback must take over again because we need the hormones within their normal level. So I just put negative feedback returns. But during the special event, positive feedback must occur. Or again, the special event can't occur. This has nothing to do with homeostasis, by the way. So positive feedback is non-homeostatic. Has it in the notes. And it's sitting right here. Negative feedback, however, is absolutely 100% homeostatic. That's how we maintain homeostasis within the body. When we lose homeostasis, that's when bad things start to happen. And that's when people, again, come to see you to get fixed. Speaking of losing negative feedback, let's talk about it. So a loss of negative feedback now. And this is bad. So we have what's called hypersecretion and hyposecretion. And so once again, the level of whatever, I'm not even going to put the X and Y axis. I know what it is now. So there's our normal level. Oops. There's our normal level. Here's our normal range. And what's going to happen here is we're going to be outside of our normal range for extended periods of time. How long? Who knows? A few weeks, years, it just depends on the condition itself. And so, this is not positive feedback. This is what we're going to call hypersecretion. We are secreting too much hormone. Or hyposecretion. We're secreting too little hormone. Again, over extended periods of time. Now, why is that happening? Because we have lost negative feedback. Not because of some special event where we needed to increase the level of a particular hormone. And if you lose negative feedback when it comes to the hormone, whatever that hormone is regulating is now then not normal. So what I'm showing you on the screen right now, this is pathophysiology. This is pathophys. What I showed on the screen with negative feedback, well, that's physiology. So that's phys right there. If somebody asked me, will you draw physiology in one picture? That's what I would draw. That's what I would draw. Because physiology is a study of when things go normal, when normal processes within the body. Where pathophysiology is a study of processes that are no longer normal. And so why would that occur? We've lost negative feedback. That's the reason. Now, why would we lose negative feedback? There's going to be many different examples when we talk about hypersecretion and hyposecretion. And that's how we're actually going to approach the hormones. With the hormones, what we're going to do is, I'm going to talk about a bunch of hormones. We're going to talk about this. We're going to talk about how we keep hormones within their normal level, their functions, and then what we're going to do is, well, what happens if too much of the hormone is released? What happens if too little of the hormone is released? And actually, we'll probably spend a little bit more time on the pathophys when it comes to the hormones. It's just more interesting to talk about, in my opinion. And again, that's what you guys are going to do someday. You're going to diagnose people with these particular conditions that we're going to be discussing in this chapter. And again, we'll do as much pathophys in this particular course as we possibly can. Again, because you're going to get a heck of a lot of it in med school. So, look at that. We're at 45 minutes. I'm going to give you a break. The break will be a few minutes long. I might run over and get more paper, because I think, oh yeah, I need more paper. So about 5 minutes, it should take me about that long to go there and back. All right, folks, thanks for your attention.